This talk was originally called Building Capacity for Genomic Medicine in the Desert. And you'll see why in a bit. But it's very much similar to what happened in Singapore more than 20 years ago in the sense that Qatar is a greenfield project. It was a country with absolutely no scientific enterprise. And over the last 20 years, everything related to biomedical and health really sprouted out of nowhere and is really leading the future. So in case you don't know, this is where Qatar is. It's right in the Middle East. It's a very small country by footprint. I think it is bigger than Singapore, though, in terms of surface area, but it's definitely not as populated. The historical population of Qatar has always been low, but it boomed in very recent years. But despite this boom, only a very small percentage of the population is Qatari, so there's a lot of people from outside of the country. And to give you a visual of this boom, this is what Qatar looked like up in the top left corner 40 years ago. So today it's transformed into downtown Qatar. So the little building you see there, the little pyramid, let me see, can I get a mouse in here? Yeah. So this is the Sheraton Hotel. It's iconic, but I dare you to find it in this photo, right? So it's basically over here. And this is what the skyline looks like. So over the last 15 years, Qatar really transformed tremendously. And this is all part of an ambitious makeover and part of the National Vision 2030. Qatar Foundation, where Education City is based, was founded to develop sustainable knowledge and a knowledge economy. And one of our major milestones right now is the readiness for the World Cup. So I hope you all will join us there in a couple of years. So a little bit about Sidra Medicine. Sidra Medicine represents the vision of Her Highness, Sheikh Hamoza, the chairperson of Qatar Foundation and of Sidra Medicine. It was developed as the first academic medical center of its kind in the Middle East. And there's a tripartite mandate of patient care, research, and education. The vision is that Sidra wants to become a beacon of learning, discovery, and exceptional care, ranked amongst the top academic medical centers in the world. And as part of the mission statement, two parts of the mission statement, the first part focuses on the clinical service, but the other two parts really relate to you and to us, which have to do with research and training the next generation of students and staff. And to give you an idea of scale, this is what Sidra looks like. So this was Sidra 10 years ago, and this is Sidra Medicine today. And we have a very beautiful statue of embryonic development. It's very unique. It's right outside of our building. Ian's seen it a few times, probably took selfies with all of the individual statues there. But this is massive by scale. And we do a lot of research. Our main focus is on rare undiagnosed disorders, because I'll talk to you a little bit later about why. We also have a program on common and immune-mediated disorders like diabetes and asthma. We have a smaller cancer and a smaller maternal fetal focus. And we are also the national core for genomics and bioinformatics for the country. So this being an academic talk, this being an academic audience, and with my own proclivities as an academic, I thought that I'd start this talk by first teaching a little bit about genetics, just to bring everybody up to speed. And then I'll tell you about genomic medicine, and then talk to you a little bit about what our lab does. And from there, I'm going to transition to what's happening in the country, and finally, a little bit of sociology, because I know that there's the departmental intersect here. So genetics 101, the central dogma of human genetics. Pardon me if this is too basic, but this is basically what we're taught in textbooks, that everything starts from DNA, DNA becomes RNA, RNA becomes protein, and sometimes in the omics era today, we call this the genome, there's a transcriptome, there's a proteome, and there's tons of omics these days. But the idea is that when you talk about human traits, how a geneticist thinks is that all traits are genetics, right? Some are more genetic than others. So there's single gene traits, there's multi-gene traits, there's genes that interact with the environment. But the idea is that a trait is not necessarily a bad or disease, right? And a geneticist focuses on disease because disease traits are just easier to identify, because disease individuals self-collect. They all come to the hospital. So it's very easy for you to collect them versus building large cohorts of different traits. But the central hypothesis is that alterations in your genes that are responsible for physiological pathways result in clinical observations called patients. And so if we can find what genes cause disease, then we can understand the physiological pathways that these genes operate in. So human disease genetics, again, this is a little bit basic, but bring people up to speed. There's two main disease areas: common disease, which have high incidence in the population, things like asthma, diabetes. They have a high degree of variability and a high degree of variability in severity and age of onset. These are likely caused by many variants, each with a small effect. And this is a comparison to rare disease. I'll talk a lot more in my talk about rare disease, so I just want to introduce what rare disease is. Rare disease is very low incidence. So things that, like, you would know someone with a common disease, you wouldn't know someone with a rare disease, but you would have heard of someone with a child with a rare disease, for example. The genetics of rare disease are usually caused by single gene mutations. The rationale is very simple, that if you have this rare mutation that causes a rare disease, the rare disease individual is unlikely to make it through reproductive fitness and unlikely to pass that variant on. And that's why these variants remain at low frequency. So this is sometimes known as a rare disease, rare variant, common disease, common variant hypothesis. So I said variant. Let me just introduce variant. Again, this is very basic. But a variant is basically you have 4 bases of DNA, and each of these letters, when it transforms from it mutates from one letter to a different letter, is called a variant. And biologically, this is what a variant does. So your reference genome might have a TTC, which would encode a lysine as a protein. And then if you introduce a variant at different places in that TTC, you end up changing the protein structure. So in one of the cases, you would end up truncating the protein, and that could cause disease. So it's important to note that variants occur spontaneously, 1 in every 100 million bases. So there's about 17 new variants per generation. And the general idea is that if the variant does not affect reproductive fitness, it will remain in the population. So this is a nice graphical look at variants. Variants that are new or deadly that cause disease will be limited to certain families only, whereas variants that are old or beneficial become common in the population. And from a genetics point of view, we look at the variant, the frequency of variants versus the effect of the variant. So things that are at very high frequency usually have very low effect, whereas things that are rare, or sometimes called Mendelian disease variants, have a very high effect. So these would cause disease binarily. So if you have this mutation, you're definitely getting the disease. Whereas here, if you have this mutation or maybe hundreds or thousands of different mutations, that might still only be a risk factor for disease. So again, rare disease is generally caused by rare variants, and common disease is caused by common variants. But variants are not just informative in disease. I know that there's people here from the Genome Asia group, and I just wanted to put a shout out for this, that variants also help us track human history. So because variants occur spontaneously everywhere in the genome, like I said, a bad mutation becomes rare due to its effect on reproductive fitness, but all other mutations, they just drift with us. So as they increase in frequency or decrease in frequency is a matter of chance. But once a variant is created, it becomes a permanent mark in your genome. There's basically, you cannot reverse a variant. So what happens is that as the populations keep splitting over history and additional variants come in your sublineage, then those get stuck with you, and that's how we can track population history. So we inherit a mixture of our ancestors' variants. So for example here, if this person would have inherited a mixture of variants that were introduced from this parent and then from previous generation and so forth and so on. And that you sometimes see these figures in talks, and this basically tells you that these haplotypes are specific to different parts of the world because the mutations that cause them occurred in that part of the world after humans migrated to that part of the world. And sometimes you see these figures, like some variants are very specific to some populations, whereas other variants are not seen in different populations. And this is just another way of looking at it in Europe. So the idea is that variants tell us about population history. They tell us about which population is a subpopulation of what population, so you can tell which population occurred first, and then which one came next, and which one came next, and that's how we track human history with variants. So I'll switch from that to genomic medicine and my definition of genomic medicine. So traditional medicine is the following, right? So people go to medical school to learn this. We are in a medical school campus now, right? And so you go to medical school and then you specialize and you become a neurologist or a cardiologist or a gynecologist or an anesthesiologist. And so you're taught to think of medicine by organ system, right? And that's how that's how traditional medicine is taught. You become a specialist in your organ system. But in reality, we know that all the organs originated from a single cell, and that single cell originated or had basically one template, one instruction book to create that single cell. And that instruction book was used to create all the organs. And so the idea is that that instruction book literally encodes what will happen to those organs. And your genome is equally alive and evolving as your organs. Today it's very measurable through next generation sequencing. And the genome transcends tissues because it existed before and it can create all the different tissues. It definitely predates disease. So it can improve both diagnosis and risk assessment. And your genome, as we're finding out today, is directing therapy. So you take drugs based on your genome is much better than taking a drug based on which organ is affected by the drug, by a disorder. So genomic medicine is really this concept that if you study the impact of genomic variants, then you would understand the risk for disease, the diagnosis and treatment, and you'd better be able to improve population health. So let's talk a little bit about what kind of research we do in our lab. So I'm interested in two key questions, and I think these are very, very, very big questions. It would take lifetime and generations, and it would be unsolvable in one lab. But these are the two questions that really drive me to wake up every morning and go to lab. First of all is what causes human disease? So what variation in our genome causes certain disorders, or what's the impact of environment and lifestyle on the chances of getting a disease? And the second big question is what background variation exists in our genome? And this is a very population-specific question because, like I said, you inherit the variants that existed in your ancestors. So that's a very population-specific feature. So what ancestral mutations do we carry, and how do they influence our molecular and phenotypic traits? So to put it, again, graphically, you have the burden of pathogenic mutations, which is question number one, framed in a population-specific variation. And together, this converges on predictive, personalized precision medicine. This is where the future of genomics is taking us. So I'm going to answer this question first because it predates chronologically, and it sets the stage for us to understand what causes disease. This is just an introduction to our cohort. We've recruited over 3,000 subjects so far. The majority of them are healthy, three-generation Qataris, from whom we've discovered quite a bit about population history. And then we've also recruited a lot of disease families. And the idea is that we have these set up in a biobank, and you can access these samples, and by applying NGS first, next generation sequencing, and then following up with transcriptomics and metabolomics, we have published studies, but I'm not covering them here. We can answer these questions. So from the healthy cohort, we answer questions such as what's the population origin? Do we need a reference genome? And what are some common variants that are in the population that might influence common disorders? And from the disease families, these are rare disease families, we discover Mendelian variation, founder mutations, and disease burden. And eventually, we can converge onto predictive, preventive, personalized medicine by framing disease mutations in the population context. So one last sort of pseudo-introductory slide. By now, you should know what next-generation sequencing is, but just in case you don't, this is a very simple way of looking at it. What next-generation sequencing does is you take a DNA sample, we chop it up into very, very, very small fragments, we read all the fragments, and then we computationally realign all the fragments to resynthesize the genomic sequence. So a basic takeaway of next-generation sequencing is that the average human has about 4 to 5 million mutations. The farther you are from Africa, the fewer mutations you have, and the more African you are, the more mutations you have. Only 1% of these mutations occur in coding genes. So a lot of people don't realize that only 1% of your genome is actually coding. It doesn't mean that the other 99% is not important. In fact, if it wasn't, then it would have disappeared with evolutionary history. But it's there, it's maintained, it's conserved. So there must be a very, very important function for it. And what people don't appreciate is that each of us carries about 100 stop mutations in our genome. So out of our 20,000 or so genes, we all carry at least one inactivated copy, and about 3 to 10 of our genes are completely knocked out. Yet we're all, almost normal. So this graph has gone around a lot. It basically tells you that the pace at which we can sequence genome is becoming much, much, much faster because the price of genome sequencing has dropped over 1 million-fold in only 10 years. And this is what really enabled us to move from sequencing 1 or 2 or a few genomes to ambitious projects like 100,000 genomes, and now going into millions of genomes around the world. So I'm going to go back to this, and let's talk a little bit about what we discovered about population history. So there's a summary of population genomics in Qatar. So about 8 years ago, we published a paper that showed that the Qatari population is largely made up of 3 ancestral subpopulations. So these again are 3-generation Qataris. So everyone who was sequenced here, these were not sequenced, these were just genotyped. But everyone who was genotyped here had all 4 grandparents were born in Qatar. So these are what we would call the indigenous population there. So the majority of them were of Bedouin origin. So these are the Arabs, the ones who were there a long time ago. And then we have a Persian, South Asian population and a much smaller African population. And the idea is that, Qatar being a peninsula, it represents both a trade route in the Arabian Gulf, but it also probably just reflects that our ancestors loved the beach. Don't blame them. So again, just to drive home the point, you're never 100% one population. So what this figure shows you is that individuals who are marked as Q1, Q2, or Q3 have different proportion of ancestry within their genome. So each of these columns is one individual. And so even if you're marked as a Q2, it's because the majority of your genome has, comes from Persia or South Asia, but you still have some ancestry that overlaps with Africa and some ancestry that overlaps with Bedouin Arabs. So you're never really 100% belonging to a certain population. And this is very important because, especially for a sociology audience, where your ethnicity and your genome and how those two interact with each other, it's very important to make this distinction. So part of what we discovered is that the Qatari population, is consistent with a bottleneck of 60 to 80,000 years ago, which is consistent with the out of Africa migration. What we know sociologically about the population is that it's patrilocal and consanguineous. So patrilocal means that if a man gets married to a woman and they want to start a family, they would usually move to the father's side of the family. So the woman would come to the father's tribe, not the other way around, or they wouldn't live independently. So that's what patrilocal means. And consanguineous means that people are marrying their first or second cousin, so they're related by blood. So to give you just a glimpse of the diversity of the population, so those who are Bedouin have Y chromosomes that are known to be very, very Middle Eastern. So these are self-identified Bedouins, and in fact their Y chromosome is highly Arab, which is consistent with that. But to show you the mitochondrial DNA, so you inherit your Y chromosome from your dad, but you inherit your mitochondria from your mom. So the mitochondrial DNA shows that even for these Bedouins, their mothers come from a much, much more diverse subpopulation. So this is consistent with patrilocal practices. And then this shows you consanguinity. So this is a graph that shows you the inbreeding coefficient or the coefficient of relatedness by subpopulation. And as you'll appreciate that Q1, or the traditionally Bedouin subpopulation, the majority of the people who we sampled here, who we sequenced, are related between first and second cousin degree. So this is a very, very, very high population coefficient of inbreeding. And this gets translated in their genome in what's called runs of homozygosity. So a run of homozygosity just means that there's a large chunks of your genome that are homozygous. So you can appreciate that Q1s have many, many, many runs of homozygosity in their genome. This is important for disease mapping, which I'll get to later. But part of the excitement that we first observed when we sequenced I should have said this was sequencing 100 individuals. Part of the excitement was that we discovered that a subpopulation of Q1 actually appears to be ancestral to all other global populations. So they're still less ancestral than Africans because humans originated in Africa, but they appear to be ancestral to all other global populations. So the idea is that, in textbooks you'd see out of Africa migration looking like this. So humans originated in Africa, some estimates now go up to 250,000 years ago, but that about 100,000 years ago humans migrated out of Africa. And the idea is that the entire world population is a subset of African submigration. But what we discovered through this Bedouin population is that some individuals made a hard right and went into the Arabian Peninsula and stayed there ever since. This is supported not just by level of diversity, but also by that subpopulation having much lower levels of Neanderthal admixture. So this has become, ancient DNA sequencing has become a really big field now. And by looking at how much Neanderthal DNA we find in an individual, we can tell where their population has passed through history. And Neanderthals, and Denisovans are the two major ancient subpopulations. And those are definitely farther away from Africa and Europe and Asia, respectively. So this discovery gave us the cover of genome research, and this was a very, very, very big achievement for us, not only to set the stage for genomics in Qatar, but because the cover of genome research that year was designed by a local Qatari artist. So I give her full credit for this. She has no background in biology or genetics or anything. We explained to her what we discovered, and then she came up with this, which I thought was very beautiful. So let me just summarize, in the interest of time, what else we have discovered in population genomics. So the Qatari founder population is old and has been relatively isolated for many years. So you get a lot of these founder mutations, and I'm going to show you some more about that in the disease genetics section next. We know by genotyping that commercial genotyping arrays perform very poorly in Qataris. So our first experience was with the Illumina 2.5 million SNP chip. So that looks at 2.5 million SNPs or variants in the genome. And 35% of the variants that I looked at were monomorphic in our population. So this means that you go out and you buy this array that looks at 2.5 million spots, but you only get 1.5 million informative markers. And this is very consistent with the concept that these genotyping arrays were designed in a very Caucasian-centric manner because those were the databases from which they were discovered. Also consistent with this is that we looked at over 1,000 Qataris to see if any of the top GWAS SNPs, so GWAS is genome-wide association studies, so if any of the top SNPs related to diabetes or obesity replicate in Qataris. So this was a very targeted study, and of the 47 SNPs that we investigated, only one of them replicated. So again, this is despite the fact that in Qatar, obesity and diabetes are at very, very elevated prevalence. I think diabetes is top 5 in the world with 17% of the population is diabetic, and obesity is around 40%. So you have this large burden of problem, but when you go and look at what's been published and you try to replicate it in this population, none of the SNPs replicate, which means that there must be different drivers, different genetic drivers that are at work here, that are yet to be discovered. And just as a final note, when we went off and sequenced the rest of the population that we had recruited, we discovered that there's about 6% of our variants are novel versus the reference genome. And what's interesting is that 15% of what's called a variant, because it's different from a reference genome, was actually observed in more than half of the people that we sequenced. So when you're a variant, you should be what's called the minor allele, which means that you shouldn't be at a frequency of 50%. But here it was the complete opposite, and so these should be referenced, and this drove us to generate our own population-specific reference. But just to drive home, this concept a little further, I'll put up this table, which shows that so this was from sequencing 1,500 individuals. You had 26 million variants, of which around 4 million occurred in the majority of the people. So these are major alleles. And then by looking at the functional category, again, I want to highlight these numbers, where about 1,000 of these variants are predicted to be loss of function. So these are at a very, very, very high frequency in a healthy cohort. And what's interesting here is that these are variants that are reported to be pathogenic. So if you go to OMIM, you go to ClinVar, there's about 2,000 variants that we observed that are reported to be pathogenic. 200 of these are present in more than half of our cohort. So there's no way these are really pathogenic. And this is just, I mean, this is consistent with other studies that have shown that these databases are, I wouldn't say fraught, but there's a lot of variants that we've inherited over time that are reported to be pathogenic, but are really cannot be pathogenic, because as we sequence more people, we're seeing them at much, much, much higher frequency in normal individuals. So let's switch from population genetics to disease genetics. Again, I'll put up this graph to drive home that, we looked at common variants earlier to look at common disease and population history. Now we're going to focus up here on Mendelian variants. So these are mutations that should be absent from the population, because if they exist in you, you should be sick, right? So this is what this graph means. They're very rare in terms of allele frequency, but the effect size is huge. So if you have this mutation, you have this disease. So Mendelian diseases can be solved with a few families, whereas common disease will require large cohorts. And then this is a very important concept, and this is a concept that has a lot of potential nowadays, is that complex diseases have tails that are monogenic. And I'll just give you an example of this. So this is, you take any physiological trait that you want, height, blood pressure, weight. Most of the people are going to fall in the middle of the bell curve, right? Statistically speaking, that's how the bell curve is designed. But then you're going to find people who are extremes. And the idea genetically is that the ones in the middle are the ones that have common variants that are influencing the trait, and then lifestyle can change where you sit. But the ones that are extreme are probably ones who have rare mutations driving that disease. And so it's very hard to classify a disease as being Mendelian or being common, and that's why many of these are complex. One perfect example of this is autoimmune diabetes, non-autoimmune diabetes. So these are all of the genes that have been related, are found to be related to diabetes. Most of these genes have variants of small effects. So these are mutations that are common, but each of them has a very low or modest effect. But then there's also a number of genes which are known to cause MODY and other rare diabetic syndromes, which have large effects. So this is a very consistent thing that you see. So you can't classify a disease as a whole as being rare or common. Most of the diseases we deal with today are complex. So the idea is that to find these, you're going to require very, very, very large cohorts. Populations like Singapore or big populations in Asia might be well-powered to do this. And Qatar, we would not be well-powered to do this because our population is very small. But to find this, all you require is a few families and a good clinician basically to find the phenotype. And this is what's our strength basically, and the strength we're playing to in Qatar. And so the way that rare diseases manifest is based on this concept of genetic isolates. So again, for the geneticist crowd, I'm sorry I keep showing very basic concepts, but I want to bring the people who are not geneticists up to speed with how we think. Right? So genetic isolate is this idea that, like I said before, each population is a subpopulation of a previous population. But the previous population might have all of these different marbles in it, right? And then every time you get a migration event, or you get a disease that sweeps through and kills a bunch of people, but only a few survive, you end up subsampling from that population. And whatever you subsample, this is called a bottleneck event, whatever you subsample becomes your surviving population, and they will grow and grow and grow in size. So whatever mutations were in these individuals are the ones that are going to persist. And all the other mutations are not going to appear in this subpopulation. So the bottleneck effect, or the founder effect, refers to the concept that all individuals in a subpopulation come from a few founders. And the founders already have a mutation load, and most of this is in recessive genes. And the population has increased homozygosity. They're easy to study because of the homogeneous environment in which they are. So good examples are the Finnish and Sardinian populations due to geographical isolation, or the Qatarites and Amish populations due to cultural isolation. And in Qatar, we have this very unique population because it's both geographically and culturally isolated. So even within the peninsula, there's sub-sociological isolation where Q1s would tend to cluster or mate more closely with Q1s and Q2s with Q2s and Q3s with Q3s. So that's why they're very, very, very suitable for Mendelian genetics. So again, the Qatari population has both geographical and cultural isolation. The family size is incredibly large. And so this ends up exposing recessive alleles because you have these families with multiple affecteds. But more importantly, this is something that people don't recognize, that when you have big families, every time you have a child, there's a risk for a de novo mutation. So you just get a larger N to look at de novo mutations, because de novo mutations occur independently of consanguinity. And so familial clustering also ends up creating these Mendelianized forms of complex disease. So like I told you, you look at the tails of the distribution, you end up clustering really rare diseases in specific families as a kind of a tribal effect. And so, to summarize it, there's a very high consanguinity rate. 23% of the population reported that their parents were first cousins, and 52% reported that they were married to their first or second cousin. So 34% of those were double first cousins, so both their parents are related from both grandparents. And if you followed next generation sequencing, you'll know that next generation sequencing was invented for this use case. It really is a very powerful tool for disease mapping in rare disease populations. This is just an example of what some of the pedigrees look like. There's usually multiple affected, there's usually consanguinity, which is denoted by double lines, and there's usually very, very big families. Again, to drive this home, academically, family pedigrees are much more powerful than unrelated patients, because in families, rare diseases become common. So families self-identify as, we have cystic fibrosis, for example, in the family, or deafness. And there's multiple affecteds, usually with the same extremely rare mutation. And when you look at a family from a medical genetics point of view, you usually look at the variants, and then by next generation sequencing, you can find dominant, recessive, X-linked, or de novo variants. So let's go back to this, and now I'm going to focus on this side of the graph. Of this figure, and look at disease families specifically. So our Mendelian disease program, we recruit families with many different phenotypes. We have these umbrella programs set up in the hospital, and any family with any phenotype can get referred. And there's two emerging themes of discovery. We have a high solve rate in known disease-causing genes, and we're also discovering novel pathways. So let me just give you a pilot study. So this has now been published, so I can talk about it. This was done with whole exome sequencing. This is very basic filtering criteria, but I just want to focus on one thing which I think is relevant to this audience. It's the fact that, how we use unaffected controls to filter down for causative mutations. And I'll show you what I mean in this next slide. So here, if you sequence, so when we sequence these families, these are the number of mutations in each child, right? That was sequenced. These are 30 kids. Okay. These are in mutations that are rare, in blue. So you get a wide range of rare mutations. So these would be the ones that you would look to first for possibility of being the disease-causing mutation. And then these are rare and likely damaging by scoring algorithms. And then the ones in yellow for each kid are rare, damaging, and affecting a highly conserved base. So usually when you're doing Mendelian genetics, these are the mutations you're looking for, because like I said, this mutation should be rare because it causes disease. But here the definition of rare is rare versus all public databases, right? So you still end up having hundreds of mutations that you have to manually sift through to find a causative disease gene. So just to give you a perspective, all public databases is looking at 250,000 individuals in all public databases. So there's still a big number that you can't really discriminate. If you add on top of this the layer of only looking at population frequency in 1500, this is what happens to each of these graphs. So you end up dropping down the number of rare disease-causing candidates to tens of mutations per individual. And this is what having population-specific references does, is it narrows down the field tremendously. Despite the fact that there's 250,000 people who have been sequenced and in public databases, it still does not even come close to matching the effect of only 1,500 population match controls. And the effect of this is much more dramatic when you look at homozygous mutations. Here we're looking at tens, And then when you add in the population matched controls, we're looking at literally in some families there's only one mutation, so you know it is a disease-causing mutation. And I think that speaks volumes about the lack of diversity in public databases, and specifically the lack of Arabs in the public databases. And so everyone we sequence, we try to contribute to public databases. So to give you an example of some of these diseases, so these are some of the families, some of the novel mutations we found in each of these families. Again, as you'll appreciate, these are multiplex families. What that means is there's multiple affected, there's consanguinity, and because they're rare diseases, they come from all sorts of clinics. And what ends up happening is that even in a small size of 30 families, you end up having this, where two separate families come in and they end up having the same exact mutation. So this is again. So here's another example of two families that came in with a nephrotic syndrome. This family had two kids. This family had one affected kid, and they both happened to share the same exact mutation. And this would be very rare anywhere in the world where there's outbreeding. But in our part of the population, by calling back the families, we realized that they were actually related to each other. And so again, this is because this mutation would have occurred in a common ancestor, in a common founder, and it would have passed down both the maternal and paternal lineage and homozygosity in each of these kids. So we're finding many, many, many examples of these. So I'm going to leave you with this, that we ended up finding a diagnostic mutation in over 70% of the first 30 families we sequenced, and we can move on from here. I wanted to mention male infertility for a very unique reason, and it's because of this idea of Mendelianized complex disease. So, like I said earlier, for all these diseases, for a disease like male infertility that affects a lot of people, how do you even begin to think about how to tackle that biologically? So, male infertility has a big genetic load, but also has a big environmental effect. So, so individuals who are here are ones who are born who are born infertile, and individuals who are here are ones who acquire infertility due to environmental exposure, for example. But like I said, all complex diseases have tails that are monogenic. So, if you take male infertility as a physiological function, then people who are here might be influenced by environment. But it's the ones who are rare, who are born, with the inability, for example, to produce sperm, are going to be the ones who probably have a monogenic switch that turned that off. So infertility affects 15% of couples worldwide. About 40 million men are affected by infertility. And a very large proportion of infertile men are completely idiopathic. So they end up going clinic to clinic to clinic, trying to understand what's wrong without a good answer. We have a very large urology department in Qatar, and so we decided to partner with them to sequence some of their families. So they have around 30 families that they identified, in which we had siblings who were infertile. And that, again, is a very good hallmark of rare disease in our population. So the idea is that if we sequence them and that we sequence a sporadic cohort, we'll be able to find new genes. And in fact, that's what we showed. These are some of the families that we have. And the hypothesis going into this is that the mutations in each family might be different, but they're eventually going to converge onto the pathway that affects spermatogenesis. So this is just a graphical representation of the families that we sequenced. We took 8 families, we sequenced them, and then we also sequenced a cohort of azoospermic men. So azoospermia is the complete absence of sperm, and oligospermia is a significant reduction of sperm, but you can still produce sperm, and then fertile men are normal fathers. And like I said, the azoospermic represent the tail of the distribution. So these are individuals who are completely unable to produce progeny. And by sequencing them, we discovered 5 novel genes that affect human infertility. Lucky for us, almost all the genes had a mouse knockout, so that really cut down the amount of work that was needed, and that's one of the interesting things about the infertility field. Statistically speaking, these are genes that survive just from a genetic point of view. When we looked at the expression of these genes in public databases like GTEx, which does tissue expression, almost all of them have very, very, very unique testis-specific expression. Sorry, this should I should give you some orientation. You can't see this down here, but this is basically the testis tissue. These are 32 different human tissues And all of them are only expressed uniquely in the testes, which is also consistent with this being a testis-specific disease. And by looking at the azoospermic men, we found additional mutations there. 4 additional people had homozygous mutations in these genes, which were absent from anyone else. And so this genetically tells you that these genes must be involved in infertility. And so our total solve rate was 14% of a cohort that was completely idiopathic to start with. So you're talking about 100 men who are infertile, who had basically no information coming into this, and we ended up giving a genetic diagnosis to 14% of them. And adding 5 new genes to literature, and I just want to point out that the infertility literature might have a lot of associated genes, but it had less than 20 causative genes. So adding 5 new genes is a significant step. And this is the kind of power that you get from going to Mendelianized forms of complex disease, by going to small populations that have a high level of consanguinity and big family sizes where you find multiple affecteds. And of course, this is where the genes work in terms of sperm production. But a hallmark that I talked to you about several times is this one gene had a very, very high frequency in the Qatari population. 0.4% is usually borderline cutoff for not rare disease causing. But because this population is so consanguineous, this is actually expected. So this is probably a founder mutation. So we did a functional study of this, and in the interest of time, I'll skip this, but this is now published. Just showing that despite the high frequency, this is a causative disease gene. So we found many, many, many variants that predispose or cause rare diseases, and this has gone into designing a Qatari chip. So we now have a population-specific disease screening chip that's being implemented in our healthcare system as a result of all of the sequencing that we've done and all the mutations that we've identified in the population. In the interest of time, I'll just skip over this very quickly. Our lab does a lot of research in human genetics, in addition to what I showed you about population history and rare disease genetics. We work on reference genomes, structural variants, and long-read sequencing. I'll just give you a glimpse of this. A reference genome is, for those of you who don't know, I told you earlier about how next-generation sequencing works. We chop your genome into small fragments, and then we align it to a reference genome. And wherever we see a difference between your own DNA and the available reference genome, we call as a mutation. So when only half of the reads that come from that part of the genome have the mutation, you're heterozygous, because the other half has the reference allele. But when all of the reads have that mutation, then you're homozygous, and that's how we end up calling your variants. But like I said earlier, when we did this in Qataris, we discovered that there's a really large number of what we call variants that are actually common in Qatar. So this argued very strongly for the need for a local reference, and we actually designed one for ourselves, and we published it. And now we're using that for all of our population calling. As I showed you earlier, that has a big impact on prioritizing mutations. So the reference genome is also fraught with gaps. So these are 2 papers that basically look at this. This was published from Iceland, where they discovered almost 4,000 chunks chunks of genome content in the Icelandic population that were completely absent from the reference genome. A much more recent analysis in Africa discovered 300 million bases that were absent in the reference genome. And so, we think of this reference as being this static thing that we can keep working against, but it really isn't, right? And so the way that we think about reference genomes is going to change entirely, and I'll show you how, in the next slide. But basically, these big chunks are what's called structural variants, and so this is just a very brief introduction. So a structural variant is basically a duplication or a deletion that occurs in your genome. And just like SNPs, where an A becomes a T or a C becomes a G, a structural variant occurs in an ancestor, and then if it's benign or if it doesn't affect reproductive fitness, it'll just keep spreading because it's just going to keep getting inherited. Whereas if it's bad for you, then it's going to cause disease, and then you probably are not going to pass it on. So this is, it could be benign or pathogenic. You would probably know structural variants by syndromes, like trisomy 21 is a very common structural variant, a very commonly known structural variant that's a complete duplication of a whole chromosome, but there's also subchromosomal structural variants. Approximately 5 to 10% of our genome is structurally variant, and in consanguineous populations, there's this added benefit of the ability to map homozygous deletions, which is this idea that you'd have natural human knockout. So you'd have these big chunks that are missing from individuals, but then when they marry their cousins and when they have kids, then you'd get kids who have homozygous deletions. And so this is sometimes known as the dispensable genome, right? The idea that the part of your genome might not be necessary for you to be normal. So we have a study, I'll show you a little bit of data on this in a few slides. But just to bring this all together, this argues for the need to do de novo assemblies and to have a new way of looking at reference genomes. And so what we're doing right now in Qatar is we're taking populations that are Q1, Q2, and Q3. We have 10 individuals from each of these populations, and we're combining Illumina short reads with linked reads and long reads to assemble long structurally resolved genomes. And we think that the future of reference genomes is going to be graph genomes rather than this static string that you align to. This is a very, very hot area of research nowadays. The last part of my talk is really about the landscape for genomic medicine in Qatar. And this is where I think that, for an audience that has never been there, or the first time you hear about it, you would have no idea what Qatar is doing, for the biomedical enterprise. But it's very similar to, in many respects, the recent Asian Cup. So, the recent Asian Cup that Qatar won, a lot of people had no idea. They were like, how did that happen? They just saw a team that came out of nowhere with no history of winning World Cups, with no history of winning major honors, and now they come in and they win the Asian Cup. And they had a pretty tough challenge going through Japan, Saudi Arabia, UAE, Korea. So it wasn't a very easy run. The idea is that everything for the World Cup started 20 years ago. You just didn't see it, right? And so that's what's going on in Qatar for genomic medicine, right? Everything started, I think genomic medicine is really the next World Cup for Qatar, and this is what's really going to sustain the economy in the future. We have 10 institutes of higher learning where we have, it's full of PhDs employed to look at the biomedical research. So CIDRA over here, and Hamad Medical Corporation are the main clinical focus. CIDRA is the main pediatric hospital. HMC is the main adult hospital in the country. And basically all the kids or all the adults will eventually go to this hospital. And then we have groups like the Qatar Biobank and Qatar Genome Project who look at volunteers. We have academic institution, HBKU, Qatar University, and Weill Cornell, which train the next generation of scientists in genomics, precision medicine, genetic counseling, bioinformatics. And then we also have institutes that are dedicated to research. So this is QCRI is the Qatar Computational Research Institute, QBRI is the Biomedical Research Institute, and the Anti-Doping Lab. And together we have national cores. And so we do have this one system where we work together. So CIDRA hosts the National Genomics and Bioinformatics Core, whereas some of the other institutes host the other cores. And so the idea to launch the ambitious Qatar Genome Programme started about 6 years ago now. It was just this big announcement that Her Highness made at the World Innovation Summit for Health. And she basically said that the Qatar Genome Project will chart a roadmap for future treatment through personalized medicine. And that single vision statement by a leader of such prominence ended up really boosting the precision medicine economy. Ended up creating CIDRA, ended up giving us a national core for sequencing and bioinformatics, ended up giving birth to the National Genome Programme. And I want to stress that the National Genome Programme is not a completely independent program. It's actually more of a governing body that has representatives from all of the local stakeholders. So they don't go and employ their own scientists. They actually use the resources in the country to make sure that there's less and less duplication and more productivity. And the vision of the Qatar Genome Programme is that Qatar will be a global leader in implementing genomic medicine and personalized care. This is just to summarize that the Qatar Genome Programme was built on 7 building blocks that include building a national health information system, having a comprehensive biobank, developing genomics infrastructure. So these are really guiding principles on which the Qatar Genome Programme was developed. So CIDRA is a set of genomics infrastructure, And most of the samples that are sequenced are from the Qatar Biobank. So let me, before I show you some of our exciting data here, let me just tell you what the Qatar Biobank does. So Qatar Biobank started about 7 years ago. And what they do is they just enroll Qataris or long-term residents. So you can go into the biobank and you self-enroll, it's completely voluntary, and in the process you're consented for research. So the Qatar Biobank aims to have about 60,000 participants over the by 2022. They now have about 25,000, and there's a wait list actually to get into the biobank of around 15,000 individuals. So there's been a lot of demand to go into the biobank. What they collect in the biobank, this is interesting for people who do trait mapping, is there's a lot of information on the individual subject. Measurements, some are very useful, height, weight, blood pressure. Some are not very useful, like grip strength. I don't know if we're ever going to find a genetic variant regulating that. But then there's they undergo this extensive clinical sort of checkup. So you sit there for about 4 hours. You get all of these measurements done. They put you on a treadmill. They get your VO2 max. They do, they measure your carotid. They do a DEXA scan for body fat measurements, and they collect all sorts of samples, saliva, urine, blood, and the samples are used or are usable for research for any of these tests. At the same time, as part of one way to attract people into the biobank is that it's almost like a free clinical checkup. So when you go there, they send a serum aliquot and a blood aliquot to the hospital, to the pathology lab, to do a full checkup for the individual. And if there's an incidental finding, a clinical incidental finding, not a genetic incidental finding, it would go back to the individual. They'll be referred to the service. For example, if their HbA1c levels are high, then they'd be referred to the endocrinology clinic for potential diabetes. So yeah, this is just a visual of what's collected. And so QGP and the other institutions are funding all the omics studies that are being done on the samples. So the genome sequencing is done at CIDRA. This is fully funded by the Qatar Genome Programme. But then we have these sub-projects where they're looking at the plasma metabolomics, RNA sequencing, proteomics. And these are all different studies that are happening. And if you're interested in any sub-project, I'd be happy to put you in touch with the people responsible for this. And the idea is to converge on this, right? Is that with multiomics, we'd be able to better understand health and disease. And as part of the Genome Program, we sat around a table and we created an academic publication strategy. And I'm going to show you some data in the next few slides on what we're doing for population structure, Mendelian diseases, and structural variation, because these are the areas that I'm working on. So for population structure, we analyzed 6,000 individuals, and we discovered that actually our naive idea of having 3 subpopulations was really naive. That that we actually have 5 populations, and Q1s can be further subdivided into Peninsula Arabs and General Arabs. Q2s can be further subdivided into Persians and South Asians, and Q3s are still one big chunk that we call African. This is a structure plot that shows you for different potential population sizes, how our population, this is the QGP column over here, compares to 1,000 Genomes Project. So these are Africans, Americans, Europeans, South Asians, and East Asians. And we think that altogether we have 8 populations. This is what discriminates our population from the rest of the world very well. This is just a zoom in of the plot showing you that at 8 populations, our QGP 6,000 individuals are very unique vis-a-vis the rest of the world. And what's interesting is that, you'll see that there's so much underrepresented diversity. This is the 1,000 Genomes Program that is supposed to represent all global populations, right? But there's basically no representation of Arabs there. So this is 6,000 individuals whom we sequence down here, and each of these individuals is almost completely different than anything that's been seen before. But you see some interesting trends. For example, this is the Q1 subpopulation that I told you was unique and ancestral to the to our part of the world. So they share no ancestry with the rest of the world. But then some of the general Arabs, and some of the other Arabs that have more global representation, you see they share ancestry with the Spanish and Tuscan populations, which is consistent with recent population history and the spread of the Islamic civilization. So from a PCA plot, you can also tell that these are very, very distinct subpopulations. These are just different ways of mapping these individuals in 3D space. And so we're looking at the same group, but we're just looking at it different angles. It's just showing you that these are distinct subpopulations. And again, it shows you these Peninsula Arabs are very ancestral. They have no unique, I mean, they're very unique. They have basically very few variants that are shared with the rest of the world. And in deep diving into their mitochondria and Y chromosome, as expected, the Peninsula Arabs and the general Arabs, they have Y chromosomes that are almost all J1 and J2. But they have mitochondria that come out from all over the world, and we're submitting this paper now, and it's going to be the biggest contribution to the literature of individuals with J1 and J2 haplotype. These are haplotypes that have never been described before of the Y chromosome. And consistent with what we know, these are runs of homozygosity, and so the PARs, the Peninsula Arabs, have very, very high runs of homozygosity. This is a 1,000 genomes project here. You can tell that even in the most homozygous individuals, runs of homozygosity go up to 30 megabases only. Whereas in our population, we have individuals who regardless of these are all Qatari, by the way, but Qatari with South Asian ancestry, Qatari with Persian ancestry, Qatari with Bedouin ancestry, and African ancestry. So these Qataris, due to endogamy and being culturally and geographically isolated, have very, very, very long runs of homozygosity. So what do we know about Mendelian disease burden? Again, we looked at these genomes to examine the prevalence of pathogenic mutations and population-specific burden for rare recessive disorders, and then we can investigate what we discover in their genome vis-a-vis the phenotypes in the biobank. This is to show you that just by pure pathogenic count, sorry, you can't read this, but so these are different gene panels and the number of variants we found in each gene panel. Which is it might be interesting to a medical crowd, but this is just to give you an example. For example, we found one homozygous individual for a mutation that causes homocystinuria, and in fact, this is the individual. These are violin plots where each individual from the different populations, this is the amount of homocysteine in their blood, and so most individuals are within the normal range, but then this one individual with a homozygous variant is completely separate, right? And this is consistent with being a tail of at the extreme of a physiological function. The same can be seen for apolipoprotein 3 deficiency. So if you have deficiency of apolipoprotein, you have lower triglycerides, so you're protected against hypercholesterolemia. And we have two individuals who are homozygous for a stop mutation, and as expected, they have very, very low levels of triglycerides. So that's a good thing to have. And so we looked at basically how these variants, we have hundreds of thousands of them, how they stratify, and as expected, they're very different by subpopulation. So each subpopulation has a different risk profile for different diseases, and as also expected, the Peninsula Arabs and the general Arabs have a much higher frequency, due to just being overrepresented and due to consanguinity. I talked to you about structural variation earlier, that these are deletions and duplications. And in 2015, we published a pilot study that looked at structural variants across the Q1, Q2, and Q3 subpopulations, and we discovered 16,000 CNV regions. What we discovered back then was that despite the fact that, Q1 and Q2 and Q3 have an equal number of structural variants, that actually Q1s had many more homozygous deletions. So these are homozygous deletions, heterozygous deletions, and these are duplications. Q1 had a higher density of homozygous deletions, so we had 180 genes in homozygous deletions, we don't need to cover this, but the idea is that consanguinity can lead to natural gene knockouts, and we can look at that in our population. And so what we did was we looked at structural variants in our 6,000 individuals. I don't need to show you this. This was just our algorithms, but we discovered 70,000 structural variants. We know there's a very high confidence set because the PCA with structural variants looks very similar to the PCA with SNPs. It also subdivides the populations accordingly, and we find very good examples of things. This is supposed to be a duplication. Again, PCSK9 duplication. This individual has high LDL cholesterol, and we have other deletions that we're discovering that are related to different phenotypes. So in the last 5 minutes, I'll just talk about some of the social and ethical considerations for genomics in the Arab world. We conducted a survey a few years ago that went out to public and healthcare professionals. And I'm also going to give you one slide on the Islamic ethics view in genomics. So the survey of the public, this is just a demographic cross-section for those who are interested in sociology aspects. We can skip that. There's like tons of slides and tons of information on this, but I just wanted to highlight some of this. At the time of the survey, most of individuals had heard about genetic tests, but it was borderline, and fewer individuals had heard of genomic tests. Some of the scientific literacy, some of the questions, for example, people share more genes with their paternal cousins than their maternal cousins. The answer is no. But so most of the people, 55% of respondents did not know the answer to that. Again, consistent with the sort of the patrilocal heritage that we inherited. And some other misconceptions, like every trait is controlled by a single gene. Majority of people thought that the answer is yes. And some attitude statements about why, whether or not they want to do genetic testing. So most people think that genetic testing does more harm than good. But there's a substantial population that thinks that the fear of the results becoming public makes them hesitant from genomic testing. And if you ask them, why should we do genetic testing, a smaller population approve for research purposes. But still, the majority do approve. Physician sentiments is also, has very interesting characteristics. The majority of physicians had heard about genetic testing and genome program, but many of them said that they're not at all ready to interpret genetic and genomic tests in their clinics, which is a very big factor. That's a barrier to implementation around the world. And the reasons for that are lack of genetic counselors, that testing will delay treatment, and that there's ethical concerns. And all of them are open to training. So, contrary to the misconception that doctors think they know everything, when it comes to genomics, they would gladly say that they'd be open to more training. If you have more information that you want, I can share the contact of the person who led this part of the study. And my final slide is really this: the Islamic ethics and the genome question. So just a summary of the standpoint, as the quest for genomics is necessary because it's part of man's universal search for knowledge. So there's a verse in the Quran that says, travel throughout the earth and see how creation was created, how he has originated the creation. And in looking at genes and looking at understanding developmental biology, you see that. From a benefit-harm assessment, I think the literature says that exclusively beneficial practices hardly exist, so there's always going to be an assessment that needs to be done. And harms can be controlled while benefits can be extended to the whole of humanity. And from an Islamic point of view, the collective benefit definitely trumps any individual risk or individual harm. And in the framework of the higher objectives of Sharia, the quest for genomics is important because it safeguards progeny, it can improve future generations, and it safeguards life because you understand better what causes disease. Whether it's permissible or not to do genomics, the basic concept for Islamic scholars is that everything is permissible unless specifically it is stated that it isn't. So this is called original permissibility. And that the new technologies and the new benefits are going to end up causing what's called maslaha mursala, which means that it causes collective and future benefits to everyone rather than specific harm. So the conclusion is that the involvement in the genetic revolution is a collective duty. It is part of the search for beneficial knowledge, and as it's part of civilization's growth and race towards improved health. So I'll leave you with this, that we have a whole study on this, on the genomics in the Gulf region and Islamic ethics, which we published as a dedicated wish report. And the person who's working on this in Qatar is Dr. Mohammed Ghali. So I hope I showed you, I covered all of these things in about 55 minutes, that's good. So I'll just summarize with this slide saying that in Qatar we have programs that do patient enrollment for disease. We have programs that do sequencing of biobanking and sequencing of normal individuals. We have all the platforms available to do future deep phenotyping. And we have a big push in digital health and understanding how medical practice can change with all this information. And in our hospitals, we're translating the discoveries from bench to bedside. And finally, there's a big focus on academia and on translating and capacity building and opportunities for master's and PhD programs across the board. And together, we're implementing precision medicine on a national scale. So this is my last slide. This is my team in Qatar. And please feel free to get in touch with me either by email or to follow me on Twitter if you want to get inspirational quotes or see how Man United is doing this year, which kind of balance each other out. So thank you.
Thank you, Khalid. We have plenty of time for questions. Grace will bring the microphone.
Thank you for your talk. Is the work and the type of population that you found is, and the point of view of how to address it is pretty impressive. I wonder, while you were talking, I was thinking about a population that has maybe some similarities in congenitality, the Mennonite people. Mm-hmm. I don't know if at the study of yours, if a similar study is made in if the same study is made in a similar population, but from very different place in the world, what kind of evolutionary information could we have in order to see if there are this mutation in some of the same part of the genomes that occur in a different places at a different time in evolution, no?
Yeah, so your question is if you look at another population isolate, yes, what kind of things can you discover?
Yes.
Is that the kind of, yeah, so yes. This is a good question. And it stems from the same concept that by using next generation sequencing, we can look at their entire genome. So we can basically, in terms of finding mutations, we'll find them. What they will tell us is, they will tell us a lot about population history. For example, what other populations of the world are ancestral to them, so where did they come from? And then also what other populations of the world might be less ancestral. I don't know what the correct word for it is, but maybe basal to them, which is basically what other populations of the world are a subset of that population. Right? And this is basically by looking at their genome, by seeing, like I said, the genetic marks that occur over evolutionary history are permanent, right? Because the idea is that you have this 3 billion string of letters, right? And you in each generation, you hit 70 spots, right? And you switch them. And so these become permanent marks. It's very unlikely that one of those spots will get hit again and get reversed. Very, very, very unlikely, right? And so each of these marks basically tells you, where this population came from and where it's going. Now, if those marks affect, random parts of the genome, that's going to be very informative for like I said, the population history and where it came from. But if it affects coding genes, that's where we can discover the burden for disease, right? And very, very likely, if a population has been isolated for a very long time, it's going to have its very own unique burden profile. Like I showed, in the cases that we've sequenced, most of our disease families have mutations that are in known genes, but they're completely novel mutations. So if you went into a diagnostic clinic and you said, here's a patient that presents with cystic fibrosis, can you check what mutation causes it? If you're in a Caucasian setting, they're going to check for a mutation called Delta 508, right? So yeah, because it's the most common one there. But in our population, you need to check for a mutation called I1234P. Right, which is a completely different mutation, but it occurs in about 5% carrier frequency in our population. So these are the kinds of things that you will never know until you actually do the work, right? And that's the kind of thing that drives us to to not only argue that we need population-specific references, because our profile will be very different than the rest of the world, but then at the same time that there is a lot of potential for translation in the clinic. Right, which I think is underappreciated because of when you look at only population history and not the rare diseases.
Thank you. And just one more. Can you comment a little bit more about the mitochondrial variations that you had versus the nuclear genome? I mean, because in the mitochondrial, what I saw is that you had many of the mutations that are shared around the world versus the nuclear genome. I don't know if you can comment a little bit of this.
Yeah, so this is very consistent with this concept of patriarchality, right? Is that most of the men, so most of the Y chromosomes that occur in the Middle East are ancestrally Middle Eastern. They were always there, right? And so because most of the men stayed there, but most of the women would come from all over the world, and then they would stay with the father's side of the family, right? And so you'd get individuals today, when you sequence them and you look at their Y chromosome and their mitochondria, their Y chromosome says that, well, if they're male, you can look at their Y chromosome, right? If they're female, then you look at their autosomal DNA. But both of those would argue that their ancestry, their paternal ancestry, is going to be from that part of the world, whereas their maternal ancestry is much, much, much more diverse, right? And so we saw that both in our pilot study of 100 and then in sequencing 6,000, we saw the same exact, trend.
Thank you very much.
Yeah.
So I'd actually like to ask a question about sort of social and ethical issues with getting your genome sequenced. Generally, in biomedical ethics, individual consent is the gold standard. If I want to donate a kidney, I can give consent. If I want to donate blood, I can give consent. But in these highly endogamous populations, if I'm in a family where there's a rare disease and I decide I want to put my genome online, and we've talked about this problem, it might not only be my individual responsibility and my individual consent that ought to be required, because I might be inadvertently disclosing the risk of family members or children, siblings. And so I was just wondering if you could talk a little bit about that and say what kind of conversations have been going on at CIDRA and how you might square that issue with the harm reduction commitment in Islamic ethics. And then on the same line, you mentioned that you don't bring back to patients incidental findings. You bring back clinical findings, but not genetic incidental findings. And I wonder, as precision medicine gets more efficient and more robust, how that line between those two is defined and how it might blur. And maybe there are even some cases where that has been debated.
Okay. Yeah. So to rephrase that question, I think the first part of the question is, if you get your genome sequenced and you decide to share it, do you need to take permission from other people who share your genome, like your siblings, because it's not really, it is yours, you're donating it, but in theory it's also half theirs. And so if you have a risk of something that they don't want disclosed, how do you deal with that? So let me ask people in the audience, how many of you have had your genome sequenced or have done like a 23andMe or something like that? Any kind of genetics? Okay, so very, very few, okay. How many of you want to have your genome sequenced? Okay, a little higher proportion. How many of you want to do that for medical reasons? Okay, so very, very small. So most of you want to do that for recreational reasons? Yeah? Fitness, sports, nutrition, all that pseudoscience. Anyways, okay, so here's a question. Away from genomics, if you had a sibling who had a disease, would you want to get tested for that disease? Yeah. Okay. How many people say yes? Right. So that's logical. If you had a parent with a disease, would you want to get tested for it? Okay. What if that disease was irreversible? Alzheimer's, for example, Huntington's. You'd still want to get tested, or you'd rather not know that, right? So again, it's a kind of a lukewarm response, because I think a lot of people haven't really thought that through, and it's a very important question, and that's not one that I could answer. I think that's a sociologist's job, is to really do the necessary legwork to try to figure that out, and I think that's going to differ from one population to the next. So, for example, in our population, there's a very high emphasis on collectivism rather than individualism. And so usually, if you're part of a family structure and you're asked to do something by your parents or by the family as a whole, you would do it, right? And so if that means that you get tested for a disease or to share some knowledge, then you would do it. The flip side of that coin is stigma, right? In highly collective societies, people stigmatize disease, right? They think of it as, this runs in my tribe, I don't want people to know about it, right? But as I think, as our attitudes towards disease increase, and as the availability of treatment increase, and we get better at treating these things, detecting them and treating them, I think that it becomes a moral obligation on us to get checked, right? Because the Islamic thought on that is that your body, your DNA, everything that you consider to be you is not yours. It's a gift to you from God, and then your soul is inside that, right? And so you need to take care of that gift. You need to treat it well. You need to make sure that it's well maintained and that it grows. Kind of like a healthy mind, healthy body type of thought. So it's your obligation to then make sure that if there is a preventive option, you should go and take it. And you should go and do it. Whether it's right or wrong, whether that decision should be influenced by parts of your family. In our part of the world, it's less of an issue because if your dad says go do it, you do it. But in other parts of the world, I think it is a very big debate. And I have no idea how it's going to get solved, right? And the second question was, can you repeat that again?
It's about the distinction between a clinical incidental finding and genetic incidental finding, how that word might shift.
So that's a very important consideration. So an incidental finding, to bring everybody up to speed, is the idea that you go into the clinic, you go into an endocrinology clinic to check for some endocrine disorder, and then it turns out that your lab tests show that you have you might have a cancer or something. So that's an incidental finding. You didn't go looking for that, but they found that in your lab tests. So in a clinical setting, a clinician would be obliged to as part of the Hippocratic Oath, would be obliged to work that up, to refer you to the right department to get followed up, and so forth and so on. In the genomic setting, the lines have been completely blurred because, first of all, because no one really, there's very big debate on what constitutes a reportable incidental finding. There's so much variation in our genome, we don't know what more than 90% of it does, right? And so we can't report on that. But there is variation in our genome that we do know. For example, the rare diseases, being a carrier or having mutations, for example, in BRCA1 and BRCA2 that could predispose to cancer. So the question is, should that be reported or not? So as a nascent genome project, what the Biobank is doing is that it hasn't taken up the obligation on itself to report that, just because the science behind that and the ability to do that would be very dilute right now, right? In the future, it might be the case. But the clinical test, because it's going to the hospital anyways, you can report that, and then the incidental finding can be worked up. But in a conversation with the Islamic scholars, who are generating all of the sort of social and legal and ethical implications of genetics, they say the complete opposite. They say, I don't care if you wanted to see it or not. If you found it, you have to go back, even if it meant re-identifying someone who was supposed to be de-identified, right? So that's the sort of the religious obligation, right? So that puts us at a moral quandary, because the IRB says you can't re-identify someone who you've de-identified, but the moral obligation Islamically says that if you found something, then you should be able to reach that person back. So we don't have a mechanism to do that now. That's why we're not doing it. But for example, with breast cancer specifically, because of the BRCA mutations, those have been, there's work between the Biobank and the National Cancer Center to try to counsel some of these individuals. So I don't know what's going to happen with that, but we're going to find out. Yeah.
Wondering if you could tell us about what impact you believe knowledge in the hands of people will do. Because
what I think is a brilliant study case that you have. You're so beautifully diverse, but do you think there could be a scenario where people say, oh, I want to more stand out with my one kind of ethnic affiliation and less so with another? And could this in the end lead actually to a society that ends up more divided? Or do you think this will bring people together?
So that's a very good question. So to repeat that question is, what do we think the impact is going to be of people having the knowledge of their genomics, right? Specifically with regards to ethnicity and society, right? And social origin. I mean, the way that ethnicity plays a part in social origin and making sure that there's no caste system and so forth and so on. So that's a very important question, and it's one that we struggle with a lot in the context of Qatar, because even though Islamically there should be no difference between someone of Bedouin origin or someone of African origin or someone of European origin, because humanity is just one big collective race, right? And that's how you should see it. Unfortunately, in the cultural tradition, there's a lot of us and them and a lot of subdivisions. And so one thing that the Genome Program sort of thought about in the beginning was that we will not return ancestry results, because they're afraid that might cause more social divisions and more difficulties, right, in the future. Now, I don't know if those fears are well placed or misplaced. I think I'm a strong believer in the democratization of knowledge, that I as an individual have the right to know anything I want to know about myself, and then how I use it is my prerogative. But it's not. We do have a very tribal structure in the Middle East. It is not just specific to Qatars, in the whole Middle East, and there is a lot of a sense of class or differentiation, sociological differentiation based on your ethnicity. So that's why the Genome Program does not report any of that. And there's also they're coming up with a volunteer report now to give back to the individual, and it's going to have pharmacogenetics, it's going to have some fitness, it's going to have some nutrition, just as sort of a return of results, and no ancestry for that reason.
How interested are people in this archaic admixture? Because you mentioned in your talk.
Yeah.
Is this something that actually, because I met a lot of people who were really, really curious about Neanderthal Denisovan admixture. Is this something you see people are interested in knowing?
I think that the population as a whole will be very removed from this. They won't understand, as much as because it's not a very scientifically, literate population. But in fact, in our population analysis, and maybe I can show you some data on this, later. We do compare our 6,000 individuals to there's the I think Lazardus 2016, which looked at 50 or so archaic DNA samples, ancient DNA samples. And we do find quite a bit of similarity between our Peninsula Arabs and other archaic hominins that exist throughout Europe and the Near East. So I think it's going to be I think the more knowledge we can bring out from here is going to be very interesting about population, migration in early years, out of Africa. But I don't think that this has captured the imagination of the public yet. It's not something that they would really relate to.
Because in principle, it's very important for people to understand how essential that peninsular population was when people went out of Africa. And that mix of people that got created there, that was then so successful they could go on and populate the world.
Yeah.
And that probably had a lot to do with how immune system was inherited, that was already in that situation. And I think it's important for people to understand, because everybody just talks out of Africa, but nobody talks about what was there when people walked out of Africa.
Yeah, no, I think you're absolutely right, and I'm very interested in population history specifically, and our own evolutionary history as a human race. And I think that there is so much to be discovered from this small data set. So we've now the first analysis that we've done was only on 6,000 individuals, and we discovered 1,500 of these Peninsula Arabs that have never been. They're not in any public database, right? In our first 100, we had about 12 of them, right? So they're a very small subset of the population. But now we're bringing in 1,000 new haplotypes that are that old and that anciently diverse, right? And there's so much to be discovered, both in terms of selection, positive selection, and negative selection that occurred back then. But I think the real cool science will be in structural variants. I really think that these individuals have this really beautiful genome that represents what the conditions were back then, that there's just so much to be done and so few. So if you want to collaborate on that, we'd be happy to collaborate on that. But I think there's so much cool science to be done out of this. Now the Genome Program has we've just finished sequencing the 18,000th sample. So by the end of this year, we're going to have 20,000 samples. And we can rerun a lot of these analyses on 20,000 samples. So I think there's just so much information that is waiting to be unlocked from that part of the world. And that part of the world is just a small proxy of what the rest of that region has, in terms of the secrets of human history that can be unlocked.